Okay, this is Chapter 11, Section 1, and this is the work of Gregor Mendel. This dashing young man is Gregor Mendel. He was born in 1822, and just some facts about him to get things going. He actually was a monk and a teacher, obviously, and what you can read here, a gardener and a mathematician. Why they call him a mathematician and why we're talking about him is he basically came up with some of the main ideas that we still use when we start talking about genetics and how all organisms inherit traits. So we're going to talk about him quite a bit here in the next few minutes, and here we go. Some of the things that he talked about and the things that he used. He took pea plants and he studied 7 different traits of pea plants. And there's a couple reasons. One reason is that pea plants are easily pollinated and they're easily manipulated because of their flowering characteristics. They can be self-pollinated. You can take the anthers and the stigma and mix them together and self-pollinate one single plant and create offspring of that. You can also cross-pollinate between 2 different plants because of their anthers and their stigma, the male and female parts of those plants. So he used those and he knew how to manipulate those parts to a plant. So again, self-pollinating, they have male and female parts, and you can also cross-pollinate them. The other reason they use pea plants, or Gregor Mendel used pea plants, is because they're very, very easy to keep track of stuff. And the one that we'll talk about a lot out of the 7 characteristics are tall versus short. The plants that he saw, they were either tall in size or they were short in size. And it's kind of an either or type thing with the 7 characteristics that he studied and looked at in the inheritance of traits with these guys. So he noticed 7 different things about them. A trait is a specific characteristic that varies from one individual to the next. So one of the traits, again, that he talked about was tall versus short. Another one of them happened to be the seed textures. One was round and one was wrinkled. And another one was the seed color. He could see whether they were yellow or green. So right there. So when he started crossing them, he noticed that there were certain types of plants that were true breeding. These plants happen to be true breeding because if you have these 2 characteristics, and I'm going to try to write up here so everybody can see. If you have a dominant letter, and that's supposed to be a T, 2 dominant letters, and you cross these with 2 other dominant T's. Remember, these T's are representations of DNA, and this is the DNA for tall in this case. If you cross these in a Punnett square, and this is not too bad actually for me, what we got to do is you have to segregate the letters again. So you put all the capital T's and the lowercase t's. So these are on the top and these are on the side. And then what you get back in the middle are, and when you reproduce these true breeding plants, you end up getting the exact same genotype again. So every one of these plants in the offspring will be 100% tall. And these are considered true breeding. So if you look at these guys, and if you were to see the phenotypes or see them, you would see that every single one of them ends up being completely tall. So the other type of true breeding plant that I want to talk about here, if I can clean this up real quick, are homozygous recessive. And those 2 types of plants, we start with 2, move this guy over here, 2 lowercase t's, and these are both homozygous recessive. So if we crossed both of these, and remember these t's are little genetic sequences or representation of the small gene. If we were to cross 2 homozygous recessive short plants, and we segregate the T's, so these guys now are right here, each one, and then we bring these guys, we segregate them, we put them here, and then we represent them, and we put them back down in here. Every single one of the offspring for these homozygous recessive ends up being 100% short. So they're considered what you see right here, true breeding. And what Mendel did is he did this cross. So if I can find my pen here again, here's what he crossed. He crossed 2 homozygous dominant for tall in the P1 generation with 2 homozygous recessive. What he got was what you see on the next one. He got a hybrid or heterozygous. Basically, both of these alleles was given to the next offspring, and they're called hybrids. So here's what Mendel ended up getting. In his P1 generation, the parents were tall and short. Remember, they were homozygous dominant for tall and homozygous recessive for short. What he ended up having was all tall, and he could only see the tall. He couldn't see the genes or the genotypes, so he just knew that they were all tall. He then crossed them again. He took the F1 generation, which is this guy right here, all the tall, the hybrids, and he crossed them again. And what he ended up getting, and kind of the anomaly and the part that is kind of neat that he figured out, is that those traits from the small somehow were hidden in the first generation, and they reappeared in that second generation, or that family 2 generation. So he actually counted up and saw those numbers of tall to short and made a hypothesis that there was a factor being passed on to that. So he started to put numbers and math to this information. So if you cross 2 heterozygous, you always get a 3-to-1 ratio, and in this case it would be 3 tall to 1 short. Now it's a possibility. Understand that if you were to do this 100 times, you would end up somewhere around a 3-to-1. That's why you see the numbers 290 or 2,976 and 1024. They're right around that 3-to-1 ratio. So what did he find out? And I'm sorry if my head's in the way. Mendel's findings. He concluded that biological inheritance is determined by factors. We know these factors now as genes, or a genetic sequence. We use letters to represent those genetic sequences when we start doing Punnett squares. So again, he, in the pea pod example, because of those 7 traits being exactly tall or short or round and wrinkled, he knew that they were controlled by 2 factors. Humans and other animals and other creatures are not that simplistic, but do know that this was the start to think about it. So again, continue with what Mendel found out. I want you to remember that this is homozygous dominant. If you got 2 dominant, or capital T's, homozygous dominant. This is called homozygous recessive. Remember, homo means the same, and the lowercase t indicates a recessive. So you got homozygous dominant, homozygous recessive. Then in the F1, you have what these produce is a heterozygous, also called a hybrid. In the P2 generation, we use 2 of these hybrids and cross them. So 2 heterozygous cross, again, as I said a moment ago, you always get a 3-to-1 ratio. So you have 1, 2, 3 tall, because the dominant T always will cover up or mask the recessive T, and 1 recessive plant being short. So again, that 3-to-1 ratio. Please note on the side that you would never indicate this. You would always put the capital T before the lowercase t, the dominant T before the recessive T. Something that you need to make sure you understand here, an allele is a different form of a gene. So for example, tall is a different gene, there's a different genetic sequence than short. While they'll be located in the same spot on chromosomes, they're still different genes. One indicates tall, one indicates short. So you can carry both. The tall will be dominant over the short in that case, and you'd have a hybrid. Green versus yellow pods, or green versus yellow seeds, those are 2 different genes that code for just different color, like seed color or pod color as indicated. There are 3 laws. The 3 laws are, the first one is segregation, and segregation is when you separate those letters when you make a Punnett square. Basically in meiosis, we separate genes that you get from your mom and genes that we get from your dad again, and we get this separate ability to have what you see here with the pod color is in that F2 generation here at the end, you could end up with this coming back because of the law of segregation. So that yellow pod that you see at the beginning, because it's true breeding, it gets hidden right here in this generation, and then it reappears because of the law of segregation, because they will separate from each other independently during meiosis. The law of independent assortment is a little bit different. Just because you have a pod color with green, and then you have a seed color it should be a kind of a yellow color, that doesn't mean you're always going to have that factor. On the same, just because you have a yellow pod and a green seed, that doesn't mean that those two traits go together. What we're trying to say with the law of independent assortment and what Mendel said is that every gene, every trait will separate independently from each other. So if you have mom and dad that have maybe black hair and brown eyes, you could end up with an offspring with blonde hair and blue eyes. So that's a possibility. Just because you have blonde hair doesn't mean you're going to get blue eyes. Just because you have dark hair doesn't always mean you're going to get dark eyes. So there are kids running around and offspring running around that they have black hair and blue eyes. That's the law of independent assortment, where two physical traits end up being separate from each other. The last part is the law of dominance. So some genes are expressed over other genes, and some alleles are dominant while others are recessive. So let me get the words up here. And basically, because of the law of dominance, and again, there's some exceptions to this, but the dominant gene will always mask, hide the recessive gene in the phenotype or the physical characteristics that you're going to see. So there's some examples of this. So anytime you have a hybrid or a heterozygous, this dominant T will be the trait seen, and we've talked about this as that dominant T equal tall. So this, if we were talking about pea plants, would be a tall pea plant. So again, the same thing, we got two dominant traits, definitely would be tall. If we had, excuse me, I meant to do this, two recessive, and this is the only way you can get a recessive trait. So this one would equal short, and you have to have both recessive genes to get short. So again, this is, I think, the best one to show the dominance. This dominant T will hide or be what we see in the offspring. So we'll see a tall plant. This recessive T ends up being what is hidden in the law of dominance. And I hope that helps for chapter number 11.